Good morning. My name is Thanga Thevar. My presentation today is about a state-of-the-art subsea holographic camera that we have recently developed. Before talking about this instrument, let me start by giving a brief introduction to digital holography. A simple digital holography setup consists of a laser and a CMOS camera that is connected to a computer. Particles that are present along the laser beam path scatter the laser light. This scattered light interfere with the unscattered beam to form an interference pattern, which is recorded by the CMOS sensor and saved in the computer. An example of the interference pattern is shown here. Applying software numerical methods, something like reverse engineering, we'll be able to recreate the particles, including the object volume, as they were during the recording process. Therefore, a single hologram records volume information that is equivalent to a stack of thousands of 2D images. We can process the hologram to pick out the useful images. This slide shows some sample plankton and sand particle images taken from processed holograms. The advantages of digital holography is clear. It can be applied to study dynamic 3D particle field for measuring size, position, velocity, and distribution of particles. Recording of volume information and extraction of focused images therein. High-resolution images to less than 10 microns. And if we record the holograms at high repetitive rate, we can create videos of individual planes. The following video shows a plankton that was recorded in such a manner. Digital holography is a technology with huge potential, but currently underexploited in subsea applications. Why this is so? Firstly, the pre-existing systems were large and heavy. The one we designed more than 10 years ago is 30 centimeter in diameter by 1 meter long and weighs more than 100 kilograms. This limits its deployment to landers and towing frames. Secondly, the hologram processing time was slow. Each hologram was taking about 5 to 10 minutes to process, not fast enough to make in-situ analysis or decisions. In this project, we have tackled both these shortcomings so that digital holography will find wider subsea applications. This image shows the holographic camera that we have just developed. It is called VHolocam. It is connected to a 50-meter long subsea cable. This will allow real-time monitoring and processing of holograms from on board a ship. This is a close-up view of the same system. Here's the holographic camera sitting here and it's connected to the subsea cable. The recording volume is between the two windows of the VHolocam. So this is a volume being recorded. The subsea cable is connected to the laptop via a control box here. The holographic camera is 9 centimeter in diameter by 60 centimeter long and weighs just 3.5 kilograms. It can be deployed on its own or mounted on an ROV or AUV. This particular camera has been designed for deployment down to 500 meter water depth. For greater depths, we just need to change the housing. For independent deployment, we have a separate battery power supply that will be connected here, replacing the subsea cable. The holograms will be saved in the internal memory and retrieved when the camera is brought to the surface. Miniaturization of the camera was one half of the project. The other half was to greatly improve the time taken for image extraction from the recorded holograms. We have used FPGA platform to achieve this. I'll demonstrate our image extraction suite with the following video. It will show consecutive processing of 20 holograms consisting of sand particles. This shows the processing of the 20 holograms, one after the other, and all the focus images being extracted. This shows the reconstruction in terms of the distance of each hologram, and that is the time below, the time taken for each hologram, and roughly the average time taken for all the holograms. So you can see the 20 holograms have taken around 1.2 seconds each for processing. There are many opportunities for the application of the developed system. With regards to marine environment, we will be field deploying the developed system in May-June period in collaboration with Marine Scotland Science. If that goes successfully, we intend to work with them on plankton time series studies. We also have collaborators in JAMSTEC, Japan, interested in studying the in situ dynamic behavior of plankton. Currently, we are working on a couple of projects to study microplastic pollution, and we'll be very happy to collaborate with other marine scientists who are here. Thank you very much for the opportunity given to present our work.
Hi everyone, I'm Cher Chow, and today I'm going to be presenting about moving to 360-degree videos for estimating fish diversity and function. And this is work that I've been developing with Maria Dornelis for the past year. I mainly study reef fish, but hopefully this presentation is going to be relevant and applicable for those of you studying fish in other systems, because my question primarily deals with linking fish traits and assemblage diversity with the functions that they deliver in their ecosystems, specifically rates of function and where their function is delivered. And so the common methods for questions such as these often involve diver surveys, and this can follow modes of focal individual follow surveys, which are great for quantifying function per individual consecutively. Or for assemblage structure, we could do visual census, and this often happens in a belt transect or a stationary point count. But the drawbacks to these diver methods is, for one, very obviously the time limit, because divers can only remain underwater for approximately an hour. And also, we are limited to one survey mode per diver because, the focus that you need for quantifying function is very different from one for species ID and enumeration. And so to get the amount of data to answer these questions would be very labor and resource intensive. And that's why the field broadly has started moving towards remote underwater videos, or RUVs. And this essentially teases apart the conventional pipeline of observing, processing, and turning that into data all in one go during one dive, to splitting that to having a video observe in the field and then conducting the processing and data conversion back in the lab. And one advantage of this is that we can conduct multiple extractions on the same video source. And this is an example of an RUV from the Great Barrier Reef that I used for my research in the past year. And as you can see, for my question, I investigated biting activity here. And that's one where I can watch for biting behaviors and then watch again for species ID and then relative abundance. And so using this method, we were able to investigate coral juvenile responses to the feeding trait diversity in these fish and detecting a relationship between coral spat and functional divergence. But there was still room to improve on these RUVs, mainly in relative abundance and the problem of recurring individuals. You can navigate this issue with either using max n, so the maximum number of fish occurring in the same frame, or a specialized protocol to help you distinguish between fish that swim out and then come back in and whether they are the same individual. And a recent paper pointed out that there could be a bias introduced when you look at different aggregation behaviors of fish, so that abundance estimates would be more conservative for those that are generally solitary compared to those that are more social and shoaling fishes. And secondly, these RUVs are not very spatially aware, so you can't georeference locations of where these functions are delivered. You can only tell that they are being delivered within your sampling radius in the field of view. And so that's where we bring in the 360 degrees, because it sort of reintroduces back advantages to diver surveys that we lost when we moved to RUVs, specifically the power of periphery. And so when we view 360-degree videos in VR viewers, it looks something like this. If my next slide can advance. And so this here is an example of a full spherical video where we can preserve the advantages of RUVs and their pragmatic ease and flexibility, but also increase the ability to estimate abundance accuracy. And with the VR 3D viewing, we can also use markers to better relate this with locations of where bites are being taken with photogrammetry models that we have of reefs that Viviana will be talking about after this presentation. Thank you, everyone, and I just wanted to acknowledge my team members, and here are my contact details if anyone would like to discuss this further with me and Maria. Thank you.
Hi all! Today I'm going to talk to you about how my lab studies coral in 3 dimensions. Here you have all the names of the people in the lab and all the funders that made the research possible. In our lab, we study the dual relationship between organisms and their environment. So not only how an organism modifies the environment with its presence, but also how they get modified by the habitat environment they build. And to do so, we focus on coral reefs and corals, which is a unique opportunity because literally coral reefs are built by coral, which then inhabit the reef. So this dual relationship is very, very tight. And to study this relationship, we focus on the 3 dimensions of reefs and corals. And to do so, we use structure from motion that allows us to have 3D digital replicas of our study system, both reefs and corals, repeatedly over time. And it just worked by stitching together a series of overlapping images, and then this algorithm gives you measurable 3D models. At the reef scale, we use a couple of different techniques to collect our overlapping images. So with big extent, we use automated underwater vehicle that capture the images we need and get the coordinate of where the picture was taken. As a result, we have high extent map of the reefs, as the one you see here, which has the size of 1 football pitch. And resolution, when you have a map, to give you an idea, every pixel is down to 2 millimeters. Another method we use is the reef record method, which is cheapest and faster. And to do so, we still rely on a manually swimmed camera system. And we just swim towards a pole attached with a cord to ensure the overlapping of the image. And so we swim in this nice spiral pattern. And this is to show you an example of the maps we get with that methods. These are, as you see, in 3D as well, and the size of these maps are circle of 12 meters of diameter, which for a size reference is half a tennis court. From our 3D models, we get our data repeatedly over time. And specifically, we get the digital elevation models of each reef we monitor that gives us the possibility to measure complexity metrics of the habitat. We get coral cover by outlining the visible colonies in the map. And we also bring these maps printed in underwater paper in situ and annotate the position of every colony that we find, so that we have spatially explicit coral species distributions we can study. And with this data, it's how we found and followed coral cover through time. An example that you can see here, it's what happened to coral cover when it underwent 2 cyclones and 2 coral bleaching events. So as you see, the coral cover went down, and then in 2018 was starting picking up. And this data also allowed us to see the relationship between habitat complexity measured as rugosity, fractal dimension, and high range, and species abundance and richness. And we found that reef portions that have specific combination of these 3 complexity traits correspond to higher values in abundance and richness. And then we studied structure also at the organismal scale. And to do so, we started by thinking about how to measure differences in shape of corals. And so what Kyle, a previous lab PhD student, did was to just go around different museums around the world and 3D scan the skeletons of the coral. And from there, he built the morphospace that coral shapes occupy and found that the main axes of variations were across top heaviness, surface complexity, which is a measure for how convolute the surface of the colony is, and volume compactness, which measures how close to a sphere a colony is. And because these metrics can be inferred from planar area, we were able to look at how the prevalence of these traits was changing across disturbances, such as the one I told to you before. But we can't use laser scan underwater. And so for that, we use structure from motion, and we just follow colony year by year and then compare them as they grow. And that was me. And I leave you with this sequence of maps. Thank you.
I'm John Halpin, and I'd like to tell you about marine image analysis and the North Sea 3D project. So by the end of this presentation, I'd like you to understand what skills I can offer you, what the North Sea 3D project is, the technology behind it, and why you should give us all of your ROV footage. So a background about me. So I've done two postdocs in high-end analytical electron microscopy at the University of Glasgow. One of my favourite image that I've taken is in the bottom left. So the little bright spots that you can see are columns of atoms, which I think is very, very cool. And then I went on to Cancer Research UK, where I worked as a biological image analyst, and that's turning images into useful numbers. So if you think either of these skills are useful for you, do come and get in touch. So onto the North Sea 3D project. So there's an increasing number of offshore structures, and these become quickly colonised and then become artificial reefs. And the role of these structures in the marine environment requires quantification by species and biomass, and this is where North Sea 3D comes in. So the North Sea 3D project will achieve this by using a combination of photogrammetry and semantic segmentation by convolutional neural networks. This will generate 3D images of our marine growth classified by taxa, just using standard ROV footage, and this will be calibrated for biomass quantification. So we're going to need to generate our 3D model. To do this, we can use a photogrammetric pipeline, which can generate 3D models for multiple overlapping images. In our case, 3D reconstructions of marine infrastructure from ROV footage. So we start by extracting keyframes from our video. We align these keyframes. It's known as structure from motion. Then we built a dense cloud, so that's just a depth map, a depth map basically. Then from that, we build a mesh and project the texture from our original images onto that mesh, giving us a nice 3D model that you can see on the right. So if we look at that model closely, what I want you to take away from this is, well, here we've got a wreck. It's the Thanga Thevar in the Moray Firth. It sank in the Second World War. So we're looking along part of its hull now. What I want you to see is that the structure, like the underwater structure, is very well reconstructed, but also the biomass on it, like we can see the kind of the fine detail in the species growing on it. So to classify the fauna, we're going to use convolutional neural networks. So a kind of a brief, kind of an oversimplified overview of how that works is we take an untrained CNN or convolutional neural network, we train it on an annotated data set, which gives us a trained CNN. Then we can take unlabeled images, feed them into that CNN and get a labelled image out, which can be projected into 3D to give us our model labelled by fauna. So in order to train the CNN, we need a ground truth. At SAMS, we've set up a platform to do this. So anyone at the institute can label images, annotate images, and then they're sent off to an expert who can kind of check them, producing a ground truth. So here's one of our kind of early examples. This was done with quite a simple machine learning sort of analysis algorithm. And you can see it's doing a really quite a good job, so that the different fauna are represented by different colours, and it's managing to pick them out quite well. So in the future, kind of areas I'd like to explore in this project are polarised light imaging. So polarised light's very good at seeing through haze underwater. Stereo cameras for doing what's known as SLAM, and also just absolute scaling of models. Multi and hyperspectral imaging, because we expect all fauna to have kind of a unique hyperspectral signature. Optimizing how we capture the images, for instance using cameras with larger sensors and stabilization and so on. Different algorithms to correct underwater imaging. There's an example on the right where we've used the Klahi algorithm. 3D scale calibration, 3D model optimization, and finally automating the whole thing. So in order to train our models, we're going to need lots and lots of underwater footage. So we need your OV footage. If you have anything that might be useful for us, please let us know. It'd be gratefully received. You might get a free 3D model of it in return. I'd like to finish up by asking for any questions and to say many thanks to Joseph Marlowe, my collaborator on the Null Sea 3D project, Lindsay Vare, Tom Wilding, who are running the project, everyone at SAMS and SAMS Enterprise, and finally to Sally Roos from Marine Scotland Science for the wreck imagery. And thank you for listening.